So thank you very much. Maybe if we could turn the lights down just in the front a little bit for a lot of these pictures. I'm going to tell you a story about some of my interests, which are in tissue fate specification. And I'm going to try to sort of tell you a story about why, just like in sports, playing defense in development is actually just as important as playing offense when you're trying to make the right particular tissue. So in Drosophila, much of the adult structures are derived from things that are called imaginal discs. So these are monolayer epithelia that are first set aside during embryogenesis. They will proliferate and get patterned during the 3 larval stages. And then during pupal development, they'll actually go through a bunch of morphogenetic movements and cell shape changes, and then you'll get the adult structures and in the end when they eclose. Okay, so I'm interested in the eye antennal imaginal disc, and that's sort of drawn for you here, or a picture of it larvally here. And this will give rise to a number of adult head structures. So it'll give rise to these large compound eyes. It'll also give rise to 3 simple eyes on the top of the head called ocelli. It will give rise both to the antennal and maxillary palp olfactory structures, which are here and here. And then there's tissue that surrounds this disc that will give rise to all of this intervening sort of head epidermis. Now, each of these tissues is controlled by a set of selector genes that will be expressed in very tissue-specific patterns. So one of the genes that we'll talk about a little bit today is ILIS. It's the Drosophila homologue of PAC6. And you're seeing it being expressed in what remains of the undifferentiated cells in the compound eye portion. But then there are genes like distalis that will be expressed in domains within the antennal field, and LIM1, which will be expressed in a region that will give rise to that head epidermis I was telling you about. And so one of the real interests is trying to figure out how do these things get segregated, and then how do those gene regulatory networks basically stay on in their segregated tissues. So one of the best sort of networks relating to the eye antennal field is the retinal determination network, which right now there are about 14 genes. And they're put into this category based on 2 really good phenotypes. One is that in loss of function mutations, you have the loss of the compound eye. And in this case, you have what looks like a replacement with head epidermis. Now, the second really amazing phenotype is one that was seen by George Halder and Walter Gehring first, in which if you overexpress any one of these 14 genes in non-ocular tissues, you can convert populations of those into ectopic eyes. Depending upon where those ectopic eyes are, you will get them to be sometimes functional if they're on the head and close to the normal eye. But at least structurally, no matter where the eyes are, they'll look just like the compound eyes. I won't take you through all 14 of these genes. There's only a few that you'll have to sort of remember in the next set of slides. Now, this particular network is evolutionarily conserved. So there is a core cassette that includes the PAX genes, the synoculus and isabsent homologues. And those are the 3 that I want you to sort of keep in mind for the rest of the talk, followed by a downstream component doxin. This core network is used throughout evolution, and it's also used not just for eye development, but for head development, musculature, skeleton, kidney development. It's used in a lot of things reiteratively. Okay, so this network can be put into, so all these genes can be put into a network, which I've kind of diagrammed here using these sort of radio buttons. Again, the main players are going to be the PAC6 genes, synoculus, and isabsent. And the idea is that together this network is pushing eye development, and what I'll try to convince you of is that it has other roles during development as well. Okay, so these undifferentiated cells in the eye disc are where all of those genes are going to be co-expressed, basically trying to get these cells to undergo an eye program and become photoreceptors, which you can see in the back half of the disc there. What I'm going to show you is that these genes also are functioning here to prevent those cells from becoming things like the head epidermis, like antennal development. And near the end, I'll show you that things are trying to prevent them from becoming very distant structures, sort of like the wing. And so the idea is that this network is playing offense by promoting eye formation, but it's also playing defense by turning off these very distant programs. And I'll try to convince you at the end that this is not just something specific to the eye, but every tissue will have to do those same types of things. So every tissue is constantly playing this tug of war back and forth. And so I'm going to start telling you about work that was done by Bonnie Wiesner, who's a long-term research associate in my lab. And her real main question was, what's the mechanism by which when you make mutants in these retinal determination networks, that the compound eye goes into head epidermis. The previous models were that all of the retinal progenitor cells that should have been there will die out, and that it's the cells, the surrounding normal head epidermis, that kind of overproliferates and fill in the field. So that was sort of the model that had existed. We had some evidence in the lab that probably that wasn't the case, and it was more likely that the retinal progenitors that were here actually undergo a homeotic switch. And I'll take you through just a few bits of data to get that across. And we're going to focus on two genes, synoculus and eyes absent. These things are expressed in nearly identical patterns within the eye disc. So they're expressed within a set of cells ahead of that morphogenic furrow, which is turning undifferentiated cells into photoreceptors. They're also expressed in all of the developing photoreceptors and non-neuronal accessory cells like the cone and pigment cells. Those are the ones that will give you the corneal nipples and also give you the red pigment to optically insulate a unit eye from each other. If you mutate those genes by knocking out eye-specific enhancers, you will lead to nearly identical phenotypes in which you have this no eye and switching to head. Now, lots of labs have shown that these two molecules form a biochemical complex. And it turns out that synoculus is a homeobox DNA binding protein, and it interacts physically with eyes absent through this 6 protein-protein interaction domain. Now, eyes absent has two known functions. It's a phosphatase, but it also is a transcriptional coactivator. And so there have been many ocular genes that have been shown to be downstream targets of that particular complex. What myself and several other labs, including Graham Martin's and Ilaria Ribe's lab and Henry Sun's lab, are showing that this complex, through a mechanism that we're still trying to work out, is there to also sit down on non-ocular selector genes and turn them off in that developing eye field. So what I'm going to show you for an eyes absent mutant really is similar for all of the mutants on that list of genes, in which, again, the no eye phenotype from the loss of function mutant. And when you're looking at the disc, as I'll show you in a little bit, is smaller. It'll undergo an apoptotic wave. This patterning where this morphogenic furrow that usually initiates at the posterior margin and marches across, transforming a sea of undifferentiated cells into photoreceptors, that never initiates in these mutants. And so the question is, why does that not happen? What happens to that entire field? So the size of the disc actually begins to matter because there is defects in cell proliferation. So if we just track in red how the wild-type disc is growing over time, and this is after we do like a 20-minute egg collection, so all the embryos are pretty synchronized, and then we age them, dissect the animals out, and then actually just figure out how big the eye field is, you can see that the eye disc grows, at a reasonable clip. Up through the first 3 days of development, these mutants look pretty normal compared to the wild type. You wouldn't be able to really physically distinguish them from a wild type disc at that stage. But as time goes on, these discs will continue to grow, but they grow at a much, much lower rate. And it turns out that that's due to two different causes. One is actually just a real reduction in cell proliferation. But the second cause seems to be that there is a wave of cell death that people have reported. So what we're looking at the top, and maybe if we could get the lights to go down a little bit more for a couple of the images, that would help. But at the top, what you're looking at is wild type, and we're staining these discs with cleaved caspase, which marks some part of the cell death. And what you can see is that there's a scattering of cell death through many of those stages, but nothing really significant. However, in the mutants, during these 12 hours, from 72 hours to 84 hours, there is this nice apoptotic wave that comes and goes. And it takes out many of the retinal progenitors. Thank you very much. But by 96 hours and onwards, the amount of cell death has dropped, but these particular tissues are still there. There's a lot of potentially retinal progenitors left. It's not that little strip of tissue around the edges. So the question is really what happens to these cells? How do they behave? And do they actually convert themselves into head epidermis? So what I'm showing you here are these later stage discs from 96 hours on, and we're looking at all the other members of that network in an eyes absent mutant. So we've mutated one member, and now we're looking to see whether the upstream factors such as PAX6, the eyeless gene is expressed, the zinc finger transcription factors T-shirt, a patterning molecule extramacrokeet, it's a basic helix loop helix protein, or even downstream members like doxin, downstream of that core cassette. If you look at any of these mutants and ask what happens to the expression of all the other genes, for the most part, all of those genes stay on in part of what remains of the eye disc. And in this particular picture, we're just trying to ask in some of these later stages where we see a little bit of cell death, not much, and we try to correlate, well, where is the cell death related to the cells that continue to express these retinal determination genes? What kind of patterns do we see? And when we do the merges, and we've done time courses on all of the different combinations, when we do these time courses, we can see, yes, there's still a little bit of cell death like you saw in the previous slides, but in the cells that actually retain those retinal determination gene expression, actually very few of those cells are dying. So you get that first initial apoptotic wave, and then whatever remains and is expressing retinal determination genes, they tend to survive and they don't tend to die out. And so the question is, what really happens to those as we go forward? And what surprisingly happened was that you will see a de-repression of all of the head epidermis genes that we know of. We tested about a dozen or 15 of them. I'm just going to show you one of them on this particular slide here, in which if we look at the type and wild type, you're seeing that ILUS is still expressed in those undifferentiated cells, and then LIM1 is relegated to that surrounding disk of head epidermis. But now if you look at the mutants, where ILUS is still remaining, as I've shown you before, now all of a sudden LIM1's expression has now been de-repressed within cells within that antennal field. So now you've got cells that are expressing both retinal determination genes and head epidermis genes. And we think overall that contributes to this homeotic switch. Now, when you're looking at whole mutants, you really can't distinguish whether there is a homeotic switch or there really has been some amount of cell proliferation, and that these cells here in the middle are really derived from the edges. So we made mosaic clones where now we can make patches of mutant tissue and ask, do they behave in the ways that we would actually predict? And so in flies, there's little tricks where you can tag mutant chromosomes with GFP or a LAC-Z marker. We're using GFP here. And then you can use flip recombinase from yeast to actually make small patches of tissue that will be GFP minus, and that should also be mutant for your tissue. So what you're looking at here are a disk in which the GFP positive cells are wild type, and any of the cells that are in black are mutant for eyes absent. And then in the bottom, you're doing the same kind of experiment, except all of the tissue that's in black is now mutant for cynoculus, the binding partner. And in both of those cases, we will see autonomously genes like LIM1, those head epidermis genes, being turned on within the mutant clones. And then here's a very common phenotype that we see in which you can get a clone in the middle of a disc, something like this, that in the adult will give rise to a cell autonomous transformation. Now, my thesis advisor, when he was a grad student, had laid out the development of the eye disc very nicely, and he measured a number of different characteristics. And one of the things that he noted was that there was really no cell migration in the disc. So coupled with the work that he had done in the early days along these mosaic clones, we can see this sort of cell autonomous transformation. Now, as I said, we've done this kind of analysis for the 12 or 15 genes that we know are necessary for head epidermis formation. So what we've been doing over the last few months is actually tracing their development in wild type and in the mutant and asking when do these genes start to get de-repressed in the eye disc. So I'll just show you two examples just to give you kind of a flavor for that. So one of the factors that we're looking at is a homeobox transcription factor called CUT that's normally expressed within the head epidermis and antennal fields. And what you can see is that it gets de-repressed pretty early, around 84 hours after development. Now, just to contrast that, we look at that gene LIM1 that we kind of focused on for a little bit in the disc and the clones. And you can see that LIM1 goes from this sort of disc-like pattern to a ring that you've seen before in the past. But now when we look at the mutants, unlike CUT, which turned on really early, LIM1 turned on quite late. So we've been trying to get a sort of a cascade of events for all of these head epidermis genes. We're trying to move into doing sort of more RNA-seq, more high throughput genomic analyses to see what are all of the genes that are changing their expressions over time, and we'll couple those with in situs. What I can tell you is that these two factors so far have a number of cynoculus binding sites. They have cynoculus chip peak sites littered throughout. So they may turn out to be reasonable targets for this complex sitting down and shutting these genes off within the eye field. And so kind of thinking about whether there is actually some conservation of this mechanism across time, there's been some really nice reports in the last couple of years showing that if you mutate the vertebrate retinal determination network in frogs and fish, you can get them to actually transiently express genes that are specific to the hypothalamus or the diencephalon and telencephalon. Now, those particular retinas won't transform into parts of the brain or the hypothalamus. Those cells will just die out. So that's a difference between flies and the vertebrate system. But at least this de-repression seems to be a sort of common mechanism. And what's interesting about the two eye fields in this particular case is that they're derived from areas that give rise to all of these surrounding structures. So we're getting sort of this localized competition where you're trying to play defense. In the vertebrate retina, you want the eye field to turn off diencephalon and telencephalon genes, and you want to turn off hypothalamic genes. And in the eye field, you want to turn off sort of head epidermis genes, antennal genes, and maxillary palp genes. So those are kinds of the correlations we're trying to make. So I want to flip this coin a little bit and talk about ectopic eye formation. So what George Halder had done was, as I mentioned, misexpress PAX6 throughout the animal, and you could get these ectopic eyes to form on the sort of the head, the various limbs, the wings, the halteres, even the genitals. This is work that we did in my lab a few years ago. And all of those 14 genes have this characteristic. Now, this particular phenomenon was thought to could be explained really easily because you're using the UAS GAL4 system, you have really strong drivers, so you're expressing the network at very high levels in a non-ocular tissue. So you're basically swamping out the system like the endogenous leg network by turning on the eye network at just very high levels. And people thought it was just a very simple switch that was just a consequence of expression levels. We took a different angle on it, trying to build upon this idea that maybe these genes are actually repressing these endogenous networks. And I think we have some evidence to suggest that one of the first steps in making an ectopic eye is actually using this RD network to shut off the leg network or the wing network, and then you can turn on the eye as a secondary consequence. So one of the experiments that Abby did, I'll just tell you about one of them, is just to remind you, eyes absent, cynoculus are expressed just within the developing eye field. So what she did was she took cynoculus just by itself, the DNA binding protein, and she expressed it within the eye field, in a domain within the eye. She didn't give it its binding partner, and it doesn't have its normal binding partner there. And she asked what would actually happen. And as others had shown, we get these ectopic eyes both in the disc and in the adult. Again, without its natural binding partner. So how does that actually occur? Is it really through an activation mechanism? Does it have its own activation domain? Or could it be through some new repressing mechanism that we're trying to propose? So what she did was try to see if it has an activation domain on its own, and then try to measure it. There we go. OK, so what she did was she did a yeast transcriptional assay where she had a LAC-Z reporter that she tried to activate by expressing cynoculus within those cells. As a control, we know that PAX6 is a very strong transcriptional activator. So it activates this reporter at extremely high levels, this sort of dark blue color. We know a couple of sister genes of cynoculus that are dedicated repressors, and they can't activate the reporter at all. Now, if you put in cynoculus, get this sort of anemic light blue color, suggesting qualitatively on its face, it's somewhat of a weak activator. But we can actually do better than that and then measure sort of more quantitatively. So we can now switch, rather than trying to activate LAC-Z, we can try to activate the histidine 3 enzyme in a yeast strain that lacks that particular enzyme itself. So we try to rescue the death of those cells by turning on this enzyme. And you can do this in the presence of an inhibitor of histidine biosynthesis. So while you're trying to overexpress this His3 gene and rescue the mutant phenotype, you're also trying to block histidine biosynthesis at the same time. So if you can turn on this gene at very high levels, you can overcome the levels of this particular activator. And you can get some sort of rough measurement of how strong this activator really is. So it turns out that eyeless over here is actually quite strong in that up to about 150 millimolar in this assay, it works quite well. Eyes absent, the normal binding partner of cynoculus is even stronger. And it can activate transcription even better than PAX6. However, if you put cynoculus in, just like we did in this particular assay, it is an extremely weak and pathetic activator and probably cannot account for the ability to turn on an entire cascade that gives rise to eye development. Now, that's still sort of a hand-waving explanation because it still can activate on its own slightly. Maybe that is really sufficient. So Abby did the following experiment where she took Cynoculus and she fused it to an extremely strong activator, VP16, to make a constitutively active form. And VP16 in all of those assays and in other assays using luciferase is even stronger than PAC6 or izapsin. So this turns this molecule into a huge activator. You can also fuse them to the engrailed repressor domain to make them incredibly strong repressors. You can express both of those molecules and ask which one can induce ectopic eyes and which ones can't. Now, those molecules, we've done all the controls to know that they're made, that they're stable, that they bind to DNA, that they function, that they do all the things that they should. And it turns out that the activating versions of these molecules have no ability to induce ectopic eyes. They can disrupt the tissue a bit, but no ectopic eye formation. It's actually the repressing versions of Cynoculus and its sister gene optics that can induce these ectopic eyes when expressed. So it somehow fits the model that you can induce ectopic eyes by actually expressing a repressor version of this molecule. So our model for how that works is the following. Oh, before I get to the model, just to show you, going back to one of these genes, I'll just show you cut, that when you overexpress the Cynoculus either by itself or the repressor versions, you can shut off cut, one of the targets that has these Cynoculus binding sites. So it may be one of the sites, one of the targets for this repression. But now let me go to the model. So the idea is that in normal eye development, you have this cascade where PAC6 is turning on Cynoculus and izapsin through the downstream network, and one of its jobs is to promote eye development, which it does quite nicely. But what I've hopefully shown you is that this group of proteins, through a mechanism that we're still trying to figure out, is also repressing things like LIM and CUT1 and all of the other head epidermis genes to prevent these guys from turning on head epidermis and antennal fates within the eye field. Now, in our ectopic eyes, you're doing something somewhat similar. When you overexpress members of this network, you wind up repressing things like CUT and LIM1, which are necessary in that tissue to make head epidermis and antennal tissue. Now, Francesca Pignoni has shown that there is a relationship between CUT and eyeless. So in the antennal field, if you remove CUT, as a default, you turn on PAC6 within the antennal field. So just as much as these genes are repressing CUT and LIM1 in the eye field, things like LIM1 and CUT are also repressing the RD network within the antennal field. So when you overexpress optics or Cynoculus, they begin to function as repressors. They turn off CUT and LIM1. This leads to a derepression of eyeless. And then eyeless now can activate these molecules just like they do in the wild type. So now these molecules are turning on eye development to make an ectopic eye, but at the same time, they're using this mechanism that we just talked about to keep these genes off so it continues as a cycle. So that's how you can really explain how you get an ectopic eye by putting in a repressor and how similar it is to normal eye development. Oh, but now what I want to do is tell you about an experiment in which we were trying to figure out whether this eye to head epidermis and eye to antennal things that we're seeing were really the limits to these tissue fates, because remember, they're all from the same disc. And I'm going to show you a set of experiments that Jane in my lab did to show you that you can actually transform the eye into to wing tissue. So what she did is the following. Okay, so we know that these PAC6 mutants give rise to a transformation to head epidermis. So she did a screen. So there's about 900 transcription factors in Drosophila. She took RNAi lines against all of them and crossed them into that mutant background to knock them out one by one in combination with PAC6. We got 9 different strains in which we turned the eye into a partial wing. And when we figured out what those were, it turned out to be 9 different components of the polycomb epigenetic repressor complex. So anything with an asterisk turns out to be things that in combination with PAC6 flipped it to an actual wing. And I'll try to prove to you that it's a wing in a second or so. But just to give you a sense of what the polycomb complex, how it works, for those of you that are not familiar with it, this faux SFMBT complex is the only complex that sits down on DNA. It will recruit polycomb repressor complex 2, which will add the H3K27 trimethylation. PRC1 will then read that trimethylation mark and add ubiquitin on H2A119. And that's thought, in one model, it's thought to prevent RNA polymerase from actually reading through. So that's how you can sort of suppress expression, gene expression. So to kind of summarize polycomb is adding these marks. It's also counteracting the marks that are added by trithorax, which are the activating marks, the opposite of polycomb. And trithorax is then, of course, doing the opposite. It's adding activating marks and trying to take off some of those repressive marks. Okay, so try to prove to you this is a wing and then give you kind of a timeline. So the I disc is the only disc in Drosophila that doesn't express any Hox genes. And so others have shown this and we've kind of tested it. I'm showing you here that there's no antennapedia expression in that disc. And so here's a developing wing, and I need you to remember the shape, that the wing looks like this funny teardrop. Now if you take that teardrop with your hand and you flip it over and you try to look at this wing, you can see that there's plenty of antennapedia expression now in that particular tissue. So we've turned down one of the early events, a Hox gene. And now I'll just take you through just a couple of pictures to show you just a few of the other selector genes that now make this into a wing. So vestigial gets expressed in a complicated pattern during wing development. In the late wing, vestigial gets expressed sort of in a stripe through the pouch. If you again take that wing and you flip it over, you can see there's that stripe of expression across the wing blade in this ectopic wing. If you look at another marker, distalis, again, that marked the antenna in the past, in these late discs, you'll see a stripe. Again, you flip the disc over, and you can see that stripe in this ectopic wing here. I'll do just one more picture to show you that it really is a wing. If you look at wingless, one of the Wnt family members, it's expressed along the DV midline of that pouch, also in a ring around what will become the future wing blade. Again, now if you flip that wing over, you're going to see there's your stripe along the DV axis, and then the expression in that ring. We've done this for maybe 20 different wing genes, and every single one of them recapitulates the pattern in the ectopic wing. Now, this turns out to be very specific to PAX6. So if you mutate other members of the retinal determination network, so let's go back to izapsin for a moment. So these two discs are ones in which we've removed izapsin, but ILIS is still present. So PAX6 is still there. And you remove polycomb at the same time. These guys they turn on a little bit of antennapedia, but you don't have a wing. If you now take in that mutant background and now also get rid of ILIS, now you start to see antennapedia grow, and you get the outgrowth, and the ectopic wing is forming. So this really seems to be specific to PAX6 as a phenomenon, and it may be a way to explain how universally expressed epigenetic factors get some tissue specificity. They may talk to one or two transcription factors that function early and are part of a gene regulatory network, and it may be very tissue dependent. And I think and just to give you the last two slides, I'm going to give you a sense of when this is happening. So I didn't take you through all of the steps of eye development, but as this morphogenic furrow moves across, it starts to initiate around the third larval instar. And what you can see is that that straight line where that furrow is has actually gone quite a bit through, and you can see a bunch of photoreceptors developing. And you see a wing being transformed from these cells here. So it suggested initially to us that you're getting a wing to transform much later in development. You would think this would be an early decision in the embryonic stages when these discs are being set aside, but it seemed to be something that was happening really late. We were kind of puzzled by that, and we took about the last 8 months to try to figure out being able to temporally control the mutant phenotypes when each of these events were occurring, and that's the last slide I'll show you here. So what we found out actually was that PAX6 and polycomb were actually required from mid-embryogenesis to the mid-second instar. You have to have those genes on at during that window in order to get this in order to suppress this transformation. You don't actually see the wing developing for quite a bit later. What you wind up seeing is the Hox gene will turn on at exactly 60 hours after an egg is laid. Then you will see events like DPP activation, apterous selector genes. Those things will turn on in between. I have question marks here because we just don't have enough animals to give you an exact hour when they come on, but they come on in that particular order. Then what you'll begin to see is you'll begin to see an outgrowth of the disc. And then you'll finally get the selector gene vestigial to turn on, and that really promotes a lot of growth. The animals will completely stop developing, and again, it's a time in which I just don't have enough animals to tell you when that happens, but the entire animal will stop developing. It'll wait for 3 days while this new wing is forming because you're trying to grow another 40,000 cells where there was only a few hundred left. So it stops for 3 days, it makes an entire wing, and then the animal proceeds through pupal development, and it goes on, and you get your adult. And so the idea is that early, you have cells that are really pluripotent, so that iDisk can actually form into very distant structures. As development goes on further, now you're more restricted to only cells that normally come from that, fates that only come from that disk. So you can flip the eye to a head epidermis, or you can flip it to an antenna. But then after some developmental commitment point, which we're trying to work out when exactly that is, then these things get locked, and now you're going to make the entire head. And so playing defense is actually as important as playing offense, if I can get my slide there. There we go. So you need to prevent all of these things from occurring as you're trying to promote your normal fate. And I just want to acknowledge the people who did the work. So I've had an outstanding group of graduate students in my lab for the entire time I've been there. These are the 5 individuals who directly contributed to this project. These are the members of my lab currently. Other members contributed to this work, to other projects in the lab, and they were fantastic as well. I have had a longstanding grant from the National Eye Institute that's funded this work, and I also need to thank everyone who paid their taxes in full. Thank you very much.